Give me 1 second. I'll be back in a second. I'm good though. How are you?
No problem. Solomon, I don't see the interpreters yet, which is
Yeah, I haven't gotten an
unusual.
email about it.
But if you can turn on at least the captioning, or maybe you already have. You have. Perfect.
Yeah, let me check the schedule.
Yeah. So Andy, thanks also. Oh, there's Jennifer.
Oh, never mind.
Um,
Yeah, the second one's unassigned, so I don't know if they I don't know if they filled it in.
Andy, thanks for helping with the CBS thing as well. That's still kind of going forward. In one of these instances where it's probably easier to just do it than to ask all the right permissions, because now we have lots of forms to fill out, which we've never done before.
Oh, geez.
But I think we're going to get there.
Good. Well, that's a great opportunity, though.
Yeah. Well, I think in interest of time, we will get started and others can just join in. So it's my pleasure today to introduce another one of the directors of the Hillman Academy, who some of you may have met. So yesterday you got to talk to Tulia, who runs the ICI site. Today you get to hear from Dr. Andy Duncan, who in addition to all of the research that he does, and he'll tell you about that today, he helps to run the entire program, and very specifically, he runs the Tech Drive, now what's called the Tech Drive X site. So it's my pleasure for those of you who haven't met Andy to introduce him to you, where he can tell you about his research and kind of his path to where he is. So Andy, I'll turn it over to you now.
Thanks David.
Yeah.
Thanks for inviting me, and it's great to see you guys. I know many of you here. There's a good TDX showing, so thank you for coming. And it's great to have everybody else here right now too. So I'm going to share my screen. So you should be able to see this. And if you guys have any questions, you can raise your hand. Sometimes I don't see that. And so you can always unmute yourself and just shout something out if you need to. And what I want to tell you today are really two stories. One of them is how I got here, what's been my path to get from where you are right now to where I am right now. And then secondly, I'll tell you a little bit about the research that we're doing in my own lab, which really has a very long history all the way from ancient Greek times until really our modern era. And I think that's kind of a neat link if you think about mythology and so forth. So just to start out then, I'm going to tell you about my journey and how I got to where I am today. So pretty much when I was a senior in high school, I lived in Eastern North Carolina in a town called Greenville. I think this is almost exactly where you guys are in your academic career. I was getting ready to start my senior year of high school. So plus or minus a year for you guys, I think. And we had this opportunity to go to the local medical school at our local university called East Carolina. They're the Pirates. And I could basically leave school, I don't know, it's like 1 o'clock every day, go to this lab, and I basically spent the afternoon there every day for the entire academic year. So I did that from basically, throughout my senior year. And I worked with this guy named Dr. Art Bodie. And he was in pathology at ECU Medical School. And they worked on a couple of different applications. One of them had to do with, hold on, let me set my pointer. All right, so yeah, one of them had to do with in military applications, how can you improve the shelf life of platelets? So basically, if you're bleeding out on the battlefield, the surgeons out there and medical personnel want to come and give you more platelets to prevent the bleeding. So they were trying to figure out how can they basically make a product that you could keep on the shelf, have a long half-life, and then use for soldiers. And then secondly, they looked at what happens to platelets when you go on a heart-lung bypass machine. It turns out that platelets get destroyed or damaged when you're on a bypass machine. So they're trying to figure out how it is that you can go about preserving their life. So that was cool, right? That was my first experience in the lab. And, you know, I played around with microscopes and made figures and learned about some questions. And probably what you guys are doing right now, although if the experiences the TDX students are having, you guys are doing a heck of a lot more than I did back then when I was a high schooler. So I think you guys are doing a great job. And then I graduated from high school, and for 2 summers, pretty much between my high school and college years, and then between my freshman and sophomore years, I went from North Carolina up to Yale University just for the summers and worked in this guy's lab. His name is Jeff Gruen. He was a pediatric neonatologist, and he was studying the hemochromatosis gene. So this is just a genetic disease. And at this time, everybody was racing to clone genes. And he was racing to clone the hemochromatosis gene. And actually, and so he was using a lot of sort of molecular biology, but also using like sequences that were on the internet, which the internet at that time in 1992 was very, very new. And it was really, to me, pretty amazing that you could have data that was somewhere up on the internet. So basically, I spent 2 summers with Jeff. That was awesome. It was a completely new research environment. And this is one thing you guys are going to start to learn as you ultimately work in different labs, is that, you know, research is the same anywhere, but everybody does it sort of differently. And so it's cool to see how different institutions and different labs ask questions and conduct themselves in different ways. So then in college, I went from Greenville, North Carolina to UNC Chapel Hill, which is a little bit further west. I was there for 4 years. And in my junior and senior years, I joined this person's lab. Her name is Pat Bakila. And in that lab, we worked on meiosis. So basically, this is how we generate sperm and eggs. And so we really looked at mechanisms by which meiosis works. So we looked at homologous chromatid pairing. And we didn't actually work on sperm and eggs. What we worked on was mushrooms. So mushrooms are a really neat model because they basically make spores. And every meiosis makes 4 spores, which are the products of that meiosis. And then we could basically go and genetically examine these different spores. So that was cool. You can see the mushrooms right here. I always joke that I can, in all honesty, tell you that I experimented with mushrooms when I was in college, which worked out pretty nicely. So I tell you, when I was in college, I thought that I was going to go to medical school, and got into doing this research, and you know, I enjoyed it. Didn't really apply to medical school by the time I got out of college, and I thought, well, what am I qualified to do? I don't really have anywhere to go. I'm not going to medical school. Hadn't applied to graduate school. And the only thing I was really sort of good at was research. So in '96, after graduating from college, I applied to some technician positions, and I joined this person's lab. Her name is Cora Jean Edgel, and was in her lab for about 3 years. So from '96 to '99. She was at UNC Chapel Hill also in the medical school there, and we worked on endothelial cells and characterized this protein called testican-1. And it was really at this point that I started contributing to some publications. So you can see these couple of papers over there. I put this one picture in right here because it's my one or two, it's an example of a brush with greatness. I don't know if you guys recognize this fella. His name is Oliver Smithies. He won the Nobel Prize in like 2007 or so, having to do with making genetically modified animals, so like knockouts and transgenic mice. So it turns out Oliver and his wife, who's right here in the middle, they had a lab just above ours at UNC, and they would come down and go out to lunch and so forth. So I didn't really hang out with him too much, but got to talk to him a few times. So that's my brush with greatness right there. At the end of 99, or somewhere around 98, 99, I decided I was tired of being a technician, so I applied to graduate school. Oh, sorry, I'm going to sprinkle these things in. Sometimes as you're going through life, stuff happens. So I got married in 1996, right after college. All right, so decided to go to graduate school and really went down the road from UNC to Duke University. Duke's another kind of powerhouse in research like UNC and Pitt. And here I joined the lab of this guy named Bob Abraham. In Bob's lab, I became essentially an immunologist. And I started to look at T-cell receptor signaling and really a lot of just signaling events, doing a lot of protein work, published a paper during this time. And typically graduate school is going to be, I don't know, the average around here is about 5.5 years from start to finish, sometimes shorter, sometimes longer. And at the end of my second year in Bob's lab, he decided to leave Duke and basically move his lab to California. And I had the choice to move to California with him or to just kind of cut bait and join a new lab. So for a variety of reasons, I decided to just cut bait and join a new lab. But everybody responds to these sort of things kind of differently, because that was like 2 years I had invested into his lab. And so to me, some people might think this is sort of a speed bump, but when it happens to you, it feels kind of earth shattering. It felt like I just drove into this big hole. So I had to kind of regroup and decided, what am I going to do? And I joined this brand new lab. So this is Dr. Tanishta Rea. She was brand new to Duke at the time. And we worked on, I was the very first person in her lab. I ordered the pipetteman, set up everything. But we started working on hematopoietic stem cells and how these are the cells that give rise to all the different lineages of your blood. And we asked how are they regulated at the molecular level? And I worked on the beta-catenin and Wnt pathway and related and the Notch pathway. This was a pretty much a fabulously successful time for me, because this lab had a lot of momentum going. And even though I lost those like 2 years from the Abraham lab, I still finished graduate school very much on time with all my peers and maybe even a little bit ahead of them. And as a result, I ended up publishing a bunch of nice papers in that lab, which really helped to springboard me to the next step. So at the end of graduate school, I decided to look for a postdoc. I'm sure you guys all have postdocs in your labs. And then also life happens. And during this time, I had 2 kids, and these birthdays are pretty much in line with where you guys are, probably closer to 2003 and 4. So, life gets more complicated when you're not in the lab, when you've got messy kids like this. So I looked at a couple of different labs that were out there, got offers to all of those, and ultimately decided to go to this lab that was in Portland, Oregon. So I moved myself and my entire family clear across the country to Portland, Oregon, to this place called Oregon Health and Science University, which is pretty much on a big hill. So you can see the medical center right here that's sort of on this hill in Portland. They have a campus that's at the bottom of the hill, and you can ride this tram all the way down over a highway. It's a pretty cool place. And in Marcus's lab, I started to work, completely switched my fields and began working on the liver. And that's what I'm going to tell you about in the second part of this talk. But started working on liver cells, and you'll actually see some of these same figures a little bit later. And here things went pretty well. Published a number of papers, which some of them had kind of a big splash. So I was able to then, at the end of about 6 years, start looking for a faculty job. when I started doing this, golly, it was 10 years ago that I started looking for a faculty position. I applied to 23 different places. Most of them never even wrote me back. The ones that did, you can see them right here, ended up interviewing and basically got offers at 3 of them, and decided to come here to the University of Pittsburgh. And so again, we go back to the map. I moved back east, getting closer to where home is for me, at least, to Pitt. And I think I started out as an assistant professor, which is kind of like your first step when you're a professor, and then recently got promoted to associate professor. So that means they're not the bosses are happy with what I'm doing. And the reason I came here to Pitt very largely was the diverse research environment. So I think you guys can all appreciate that Pitt is huge. There are a lot of good people here doing really great work in research. And the real sort of highlight to me is that there's a large liver community here. So there are a lot of people who are doing liver research, and it's all, so we have like a liver center and meetings every week. And it's kind of like going to a national or international meeting just with our people here, because the liver community is so rich and diverse that there's a lot of stuff out there that's close by that we can take advantage of, expertise and techniques and things like that. So with that, I'm just going to say this is what my kids look like right now. They're not quite as messy as they used to be. And this is actually an older version of lab. Some of those people have already left. But you basically start to create your family at home and your family in the lab. And I think you guys are all part of the families in your lab right now. All right, so that's kind of how I got to where I am today. And I think everybody has a different path that they take. And sometimes things are intentional. Sometimes things happen because they happen, like when I had to switch labs in graduate school. And it felt pretty traumatic at the time, but it ended up working out just fine. So you just sort of have to go with the flow on that. You guys got any questions on that? All right. Now we're going to just switch and talk about liver regeneration. Okay, so that's what I'm working on now in my own lab. And then liver regeneration has been really pretty well appreciated since ancient Greek times, as we like to say. So I'm going to tell you just sort of 3 little short stories here. Firstly is liver function and disease, a little bit of background. I'm going to tell you about the way the liver is unique in terms of the cells have chromosomal variations called polyploidy and aneuploidy that make the cells in the liver very, very unique relative to all the other cells in your body. And then lastly, I'm going to talk about how some of these chromosomal variations can affect the liver's function. So this is the liver. It's the largest solid organ in your body, and it does a wide variety of things. It's kind of like this warehouse. So it can make protein and glucose. It secretes bile, which is basically a detergent, and it can help digest your food. It stores all kinds of metabolites. And the other thing the liver does is it detoxifies stuff. So if you're taking medicines or drugs or alcohol, liver is really the first line of defense, where it tries to convert those things into alternate forms. So it can detoxify or convert xenobiotics. So the liver actually has this amazing capacity for regeneration, and usually people have heard about this before. So I'm showing you a full-size mouse liver that somebody cut out of a mouse. And what's unique about a liver is that you can, in a mouse or in a human or a rat, you can go into the abdomen and surgically remove a large chunk of it. So in rodents, we can remove 2/3 of the liver. You basically just go cut it off. And then amazingly, within about a week, or week 1 to 2 weeks, it'll grow back. So if you look at my hand here, a liver has 5 lobes in rodents. If you chop off these 3, then what happens is these two that are remaining basically enlarge and can grow to basically make up for that mass that was missing. So liver has this amazing capacity to regenerate. Very similar thing happens in humans, although it takes a little bit longer. So here's where we go to ancient Greek times. Does anybody know the story of Prometheus? All right, Ryan, you want to tell us, you know anything about what he does? What happens to Prometheus?
Well, after he does like a bunch of stuff, he gets sentenced to eternal torture by getting his liver get eaten by a bird on a rock for eternity.
Yeah, absolutely. Thank you. Thank you. So Prometheus, yeah, I wrote it right here. So he stole fire from the gods, and as a punishment, so he was chained to a mountain, and then basically an eagle comes to him every night, kind of rips open his abdomen and eats his liver. And then miraculously, by the next day, his liver has regenerated and he's totally fine. And then that very next night, the liver comes again and eats the liver. So this happens throughout eternity. So it's sort of this idea that the liver is constantly regenerating. You can remove most of it, and it'll grow right back. So the idea of liver regeneration has really been around for a long time. These are some pictures of different liver diseases. So this is a human liver that's been taken out of a patient. And then healthy liver, just macroscopically, you can appreciate that it's kind of a nice color, kind of this pinky color, brownish color, and it's very smooth. And you hear about these liver diseases. This is called liver cirrhosis, where you start to get kind of a bumpy texture right here, and it's associated with scarring of the liver. And then if cirrhosis goes on long enough, then it can develop into liver cancer. And one of the things that's the liver always does is the liver's, since it's involved, like the first line, like I said, at looking at dealing with drugs that come into your body and so forth, the liver is always getting a little bit damaged and then it heals itself. And so the healthy liver can regenerate just fine. But one of the things that happens as you have cirrhosis and liver cancer is that the liver fails to regenerate. It's basically incapable of fixing itself, which is one of the reasons you can get sick and die. So one of the things we want to figure out how to do is to take these pathways and things that promote regeneration and figure out if you can then take one of these sick livers and force it to regenerate. Could you use that as some kind of a therapy for when the liver is starting to fail? And that's exactly what I just said right here. So how can we figure out to activate the regenerative capacity to cure liver diseases? All right, so that's liver function and disease. Now let me tell you about this bizarre feature of the liver. So if you just focus on the image I'm showing you on the right, so what you can tell right here, this is kind of an adult mouse liver. The green things are cell boundaries, the blue things are nuclei. Does anybody see anything here that strikes them as maybe not how it should be? Does anything look just weird?
It looks like there's two nuclei.
Yeah, great. Who said that?
Madeline.
Oh, Madeline, thanks. Yeah, so that's it. So this is a hepatocyte. The hepatocytes are the main cells in your nucleus. This hepatocyte, main cells in your liver, has a single nucleus. Here's a hepatocyte with a single nucleus, but here's one with two nuclei. So they're kind of touching in the middle. It looks to me like a snowman. Here's another one that has two nuclei. Here's another one with two nuclei. They're all over the place. And you can, that's not normal. In most tissues in your body, you have a single nucleus. And one of the things that we can do is digest this liver with enzymes and get live cells out of it. And if we let them attach to a dish, you can see them right here. So this is one of these hepatocytes with two nuclei. I would call it a binucleate hepatocyte. Here is a hepatocyte with one nucleus, or a mononucleate hepatocyte. And you can see they're all over. Here's two binucleates that are kind of touching each other in the middle. So they're very, very common. if you were to determine the percentage, it's about 70 or 80% of hepatocytes are binucleate. And what we're really referring to here is something called polyploidy. So polyploidy refers to a chromosomal number that's more than 2 of the haploid number. Okay, that's sort of complicated. So diploids in your body have 46 chromosomes, diploid cells. Your gametes, the sperm and the eggs, have half that, so 23 chromosomes. And what polyploidy basically means is a doubling of this. So double 46, it goes to 92 chromosomes or 184 chromosomes. And sort of the generic term we can use is called polyploid, that can capture tetraploid and octoploid, or you can call them very specific terms. In the liver, it turns out polyploidy is driven by 2 things. So we already kind of saw this in that tissue section. So you can either have 1 nucleus or 2 nuclei. Occasionally you have 3 or 4 nuclei, but they're quite rare. So number of nuclei per cell drives the degree of polyploidy, but also each nucleus can have different amounts of DNA packed into it. So here, this is a 2N nucleus that's got 46 chromosomes. Both of those have 46 chromosomes. But look at that. Here's a single nucleus hepatocyte, or a mononucleate hepatocyte, that has a 4N nucleus. So it's got 92 chromosomes packed into there. Here's an 8N nucleus that has 184 chromosomes packed into it. So there's really a lot of diversity in terms of chromosome content within these hepatocytes. How common are they? If you just look right over here at this adult mouse liver, and what I'm showing you here is, this is a cell cycle profile using flow cytometry, which maybe some of you guys have been looking at. But the way you would interpret this is to say that about 10% of the hepatocytes are diploid. About 80% or so are tetraploid, and about 10 or 20% are octoploid. So this is really kind of weird to even wrap your mind around. It says that only 10% of hepatocytes are diploid, like most of the cells in your body. And then that's mouse. If we come over here and look at humans, what you'll see, you can, and I just pulled this figure out of the literature, but you've got age on the x-axis down here, and the y-axis is just relative number of cells. And you'll see that as we age, the number or percentage of diploids decreases, while the percentage of these polyploids goes up. So pretty similar to what you find in the mouse. People really have seen these polyploid hepatocytes for well over 100 years. And it's only been really in the last 10 to 15 years that we started to appreciate what they're doing. So there are a number of different hypotheses about what the polyploids are doing in healthy livers, which we don't really have to go through all those things right now. And there's some evidence to say that maybe they're playing a role in disease livers, but again, we don't have to go through those right this second. So when I told you my life story, but when I moved from Duke to Portland, Oregon at Oregon Health and Science University, I started to work on the liver. And one of the first things I decided to do there, well, I decided it, and then my boss said, hey, I'd like you to do this. So it was sort of both of us agreeing this was the first experiment to do. I decided to ask, what are these polyploid hepatocytes doing in the body? And people had for a long time thought that they were just terminally differentiated. And what that means is that they're in the liver and they just kind of sit there and do their job. They crank out new proteins, or they're doing metabolism, or the detoxifying things, all those different functions I talked about at the beginning. But people thought that they would not have any regenerative capacity. And we didn't really think that idea was right, so we decided to test it. And I decided to work on octoploid hepatocytes. So they were kind of like the most highly polyploid hepatocytes that we could get. So I isolated these things, a very pure population, by flow cytometry, which was sort of a technical challenge, but we were able to do. So we get these polyploid hepatocytes, and then I transplant them into this mouse. So this mouse is basically one that can get a liver disease, and its liver gets sick, and if it's too sick, the mouse will die. And when you do this kind of experiment, normally the cells that you put in, these donor cells, they have an advantage because they're not sick And when this liver gets sick, the donor cells can start to proliferate and basically repopulate the liver. So we asked the question, if I put these highly polyploid hepatocytes into the sickly mouse, will they repopulate the liver? And three things happened. First of all, the polyploid hepatocytes were very highly regenerative. So I could put in about 30,000 of these octoploid hepatocytes, and they would make 30 million hepatocytes. Okay, so they could regenerate just fine. But then something went particularly weird. So we knew that if we put in the 8N hepatocytes, or the octoploids, as they were regenerating, they made more octoploids. They could actually increase their ploidy, which we expected. The really bizarre thing is that their ploidy, as they were regenerating, went backwards. So they made tetraploid hepatocytes and they made diploid hepatocytes. So nowhere else has anyone really characterized the fact that a somatic or an adult type of cell can reverse its ploidy. The place where you see that happening is in meiosis, where you have diploid cells that can give rise to sperm and eggs, which are haploid. So this was pretty unique. And then it got even crazier, where the hepatocytes, as they were regenerating, sometimes they would gain a chromosome. So I said 2N plus a chromosome or 2N minus a chromosome. Same thing for the tetraploids and the octoploids. This is called aneuploidy. So they were sort of shuffling their chromosomes around. I told you before that polyploidy is really an increase in the number of sets of chromosomes. So 46 to like 92 chromosomes or 184. Well, aneuploidy means something slightly different. It means maybe if you're diploid and you've got 46 chromosomes, you might lose one chromosome or gain a chromosome, or lose one, gain two, something like that. So it's more of a discrete gain or loss of chromosomes as opposed to a doubling of the number of chromosomes. So polyploidy and aneuploidy are different. And it turns out, I started to characterize the degree of aneuploidy that was in mouse livers, and it was quite high. We found that somewhere around 70% of just normal hepatocytes had chromosomal gains and losses, which is what I'm showing you over here. And then this sort of complicated looking graph just says that the type of aneuploidy we found was completely random. It seemed that sometimes you'd lose chromosome 3, sometimes you'd gain chromosome 5, and so on. Every chromosome was pretty evenly affected. And we saw a very similar thing in humans as well. Research is not without controversy. So I went around for years talking about we have about 60 or 70% of aneuploidy in livers. And then there was actually a number of groups that have come out in recent years and said, we're a little bit wrong. They think it's about 10 times less using very different techniques. And so really the question is up in the air right now. Is the degree of aneuploidy in a mouse or human liver 5% or 50%? And that's something that we could debate all we wanted, and I could tell you how our technique is better, has advantages and disadvantages compared to these more recent techniques. But the bottom line is, even if you have 5% of your hepatocytes are aneuploid, that's still a lot of hepatocytes sitting in your body. So this is what I think our liver looks like. Humans have 46 chromosomes. I'm showing you mouse chromosome numbers here. Mice have 40 chromosomes in a diploid state. So we've got diploid hepatocytes, we've got tetraploid hepatocytes, and octoploid hepatocytes. We know that. And then in addition, we have sometimes you get the normal set of chromosomes, like this red one, this red diploid cell right here that has 40 chromosomes. But you can also have aneuploid hepatocytes, like the green one or the brown one or the blue one, that have some combination of gained and lost chromosomes. And you've got these aneuploid cells in the tetraploid set and the octoploid set. So within the mouse liver and within our liver, there's a tremendous amount of diversity in terms of polyploidy, but in terms of aneuploidy as well. All right, so that's diversity sitting in your liver. And then in this last bit, what I want to tell you about is what are these aneuploid hepatocytes doing? Are they doing anything to help or to hurt regeneration. So to just sort of frame this discussion, this is how I see our livers developing over our lifetime. Very early in life, our livers are all normal and diploid. So I'm showing you the hepatocytes here with a single nucleus. And then something happens, which we could talk about later, that allows them to become polyploid. So you start to see these hepatocytes with two nuclei, the binucleates. So kind of the liver's a mixture of the diploid and the polyploid livers. And then as we start to age, chromosome segregation errors happen, and we get these hepatocytes that are aneuploid. So the blue ones and the green ones and the red ones and so forth. And sometimes you got the black ones too that are completely normal. So our question is, what are these aneuploid hepatocytes doing? Are they causing disease? Are they doing anything good? You know, what's happening? And the temptation typically is to say hepatocyte aneuploidy gives rise to cancer. There's this entire field, and maybe some of you guys here are working on it, that has to do with chromosomal instability and how cells can become dysregulated and ultimately start to have unrestrained growth, giving rise to cancer. So in the liver, this is called hepatocellular carcinoma. Well, it turns out there's not a lot of good evidence to say that pre-existing aneuploidy is giving rise to liver tumors, although this is something we're exploring right now. But the other thing is maybe this aneuploidy can do a good thing. Maybe what can happen is that you've got gain or loss of a chromosome just sitting in your liver, and then when there's some form of injury, it might promote sort of some kind of adaptation to the injury. And I'm going to show you how that works, because that's exactly what's happening. So here we work on a disease called tyrosinemia. So tyrosinemia, or hereditary tyrosinemia type 2, is an inborn error of metabolism that affects pediatric patients. And we can also model this in mice, okay? And I have to show you a metabolic pathway. You don't have to really remember this, but I'm going to show you 4 copies of this and highlight a couple of different genes. So this is what happens in a normal mouse and a normal person. You have tyrosine that is broken down through this series of enzymes, TAT, HPD, and so forth, all the way down into these 2 different components called fumarate and acetoacetate. This is the healthy situation. We're doing this all the time. If you lose this gene right here called FAH, and this happens in, we can do this in a mouse, but this is also in patients with hereditary tyrosinemia. So they don't have FAH, and as a result, they get a buildup of this protein right here called fumarate acetoacetate. When that happens, it leads to some toxic events that causes the liver, the cells to get sick and die. Okay, so this is the bad thing. But what we can do is basically treat the patients or the mice with a drug called NTBC. So NTBC blocks the pathway right here. And that's okay. It's okay if you actually have tyrosine that gets converted all the way to this 4-hydroxyphenylpyruvate, and it gets stuck right there. That does not really have negative consequences. The good thing is you don't get the buildup of this bad guy right here, the fumarate acetoacetate. All right, so this is the healthy situation. Alternatively, you can maybe remove this other gene called HGD. If you remove that, then you are still blocking the pathway upstream of this fumarate acetoacetate, and so you're not having those negative consequences. So you can basically, even in both of these cases, no FAH, no FAH, and then we can just block the pathway kind of towards the top, and the mice or the people are healthy. All right, so I'm going to refer back to this as we go along. So here was the experiment. We took these mice that did not have FAH, so they're FAH mutant, and then they only had 1 copy per diploid genome. They were heterozygous for HGD, so HD is right here. Okay, so they don't have FAH, and these mice should get sick and die. And that's exactly what happens. So a lot of them die, but it turns out there are some survivors. And the survivors have a very unique feature. What I'm showing you here is a graph of body weight, and body weight's a pretty good readout for how the mice are feeling. So if they're feeling pretty good, they eat, their body weight is stable. But if they start to feel sickly, kind of like you, when you're sick, you don't really feel like eating too much, the body weight can drop. So in these mice that I'm talking about up here, the FAH mutant HGD heterozygous mice, body weight goes down, down, down. And what I don't show here are the ones that actually died, where the body weight went down, 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 down all the way. But a subset of them, basically the body weight recovered. And when I took out the livers, they looked like this. They were pale and sickly, but they had these red, healthy looking nodules or clusters. And so these things are actually regenerating nodules. So for some reason, the liver had started to regenerate itself. And the only way I could figure this happened is they started out with one copy of HGD, so they must have lost that second copy, which basically made them able to regenerate. So we asked this question, why is that? Well, it turns out HGD is located on chromosome 16. And remember in this experiment, I had heterozygous for chromosome for HGD. So they already had one inactive copy. And I thought, well, maybe the aneuploidy that we're talking about is playing a role. So maybe those cells that were starting to regenerate, maybe they actually looked like this. Maybe they had lost that wild type copy of HGD, so they were completely negative for HGD. So it would look sort of like this. Sitting in the liver, right, before we had any kind of injury, you just have these blue hepatocytes that are aneuploid, and they had lost chromosome 16. They had lost that healthy copy of chromosome 16. And then when we applied the stress to it, the one that made them sick, the cells that looked like this, that lost all of HGD, they had an advantage and could start to repopulate. And I think like this is what those little red nodules look like on the other page. Like these guys right here, they're brown. I think, oops, sorry, that's these. So we asked that question. Was this adaptive response driven by aneuploid hepatocytes? So I repeated this experiment. We generated we induced liver disease. We got some of those mice to basically spontaneously repopulate. And then we asked, what do their chromosomes look like? And I think, hold on a second. All right, I looked at this a couple of different ways, and probably the easiest way is to look right here. So this is using a genomic technique that people don't do so much now. Now it's more like a DNA-seq technique. This was called comparative genomic hybridization. And what this shows is that we look at all the chromosomes. So I'm showing you 4 mice that were repopulated, and you can see from chromosome 1 all the way to 19, and then chromosomes X and Y. And if you see this dark line kind of in the middle, that means the chromosomes aren't really changing until you get all the way to this chromosome 16. And when the line goes to the bottom, it means it's lost. So in all of these mice that had the spontaneous repopulation, the common feature they had was loss of chromosome 16, which is exactly what we had predicted. So what I think was happening is you had this uninjured liver that was pretty diverse. It had all this aneuploidy in there. And then we had these blue hepatocytes that lost chromosome 16, and they basically had an advantage when in the face of this tyrosinemia injury. And they had an ability to just start to proliferate and repopulate the liver. So we essentially get this signature of tyrosinemia resistance in this model, which had to do with aneuploidy or monosomy of chromosome 16. So to just sort of conclude this part, what we find is that aneuploidy that's sitting in the liver is able to drive an adaptive response. So this aneuploidy is actually doing a really good thing. It's cute that we can do this in mice, but does this play a role in humans? And this is an example of a liver a picture that I took out of the literature. This is a human liver that it has a lot of cirrhosis. So remember when I showed you those 3 images of human livers, the first one was a healthy liver. The second one was a cirrhotic liver. It had all those little bumps all over it. Well, what those bumps are these little nodules, and you can see them all over. It turns out about 1/3 of those little nodules or bumps are derived from a common cell. And we think that those cells, which are the M's right here, they're called monoclonal. We think those are may be derived from aneuploid hepatocytes that had an advantage. So we're in the point right now where we're trying to isolate these monoclonal nodules from human livers and characterize the aneuploidy that's sitting in there. But we don't really have any data to talk about right now, other than that we can identify these things. So moving forward, we hope to figure out whether aneuploidy is playing a role in the human liver or not. So just oh, I should have shown you this picture when I was saying all that. You know, is aneuploidy playing a role in this regenerative response? And that's what we're really aiming to determine. So with that, I'll just say that you know, a lot of people help us with this work. There's certainly lab members who are doing this work, but we also have a lot of collaborators who are out there that help us with things that we don't know very well, and they share reagents with us and techniques and expertise. And then, as I'm sure you guys are now aware, this all requires a lot of money, and we get funding from a number of different sources. So with that, I'm going to thank you for listening, and I'll be happy to take any questions.
Any questions?
I actually had a question about the liver regeneration.
Yep.
So the part that you cut off, can you regrow that part as well, or can you only grow like the parts that you didn't cut off? If that makes sense.
Yeah. So you know, you can do things like that with like starfish, I think, where you can chop a starfish in half and it will basically make 2 new starfish. I think. Liver, you can't really do that. And I think the major reason is the part that you cut off does not then have a vascular supply. So it's not getting blood and it's not connected to anything for nutrients. That's interesting. Like if you were to chop that off, oh, well, hold on. The answer to this is yes, that'll happen. So one of the things that people can do in the clinics is if I have a failing liver, I might need a liver transplant. Okay, and you know, they typically you can get cadaveric livers. So there'll be a liver donor, someone who died, and if we were an organ match, I could potentially get their liver, right? Well, that turns out the number of donor livers and donor organs in general are many fewer than there are people who need them. So that's a major problem. There just aren't enough donor organs to go around. So one of the things that you can do with some organs, and especially the liver, is something called living donor transplant. So typically this works within families where, say, I needed a new liver, and then, my brother, for example, had a healthy liver. So they could go into him, if we were the proper match, and chop off part of his liver and then graft that into me. And sure enough, there they would connect it to a proper vascular supply, and it can actually go and grow to the right amount of space. So we don't really do that experiment in rodents, but certainly in people that can help, that can work. So that was a great question.
Thank you.
Yep.
Any other questions? Feel free to put them in chat, unmute, whatever you would like.
Anybody buy it that this liver regeneration has been going on since ancient Greek times? There are a lot of similarities. I talked a little bit about liver cancer and I talked more about liver regeneration, but some of the things, there are a lot of similarities between regeneration and cancer in the liver. So the liver has this ability to grow more of itself. And so there are proteins and molecular pathways that turn on when the liver has to regenerate. And that's a good thing. But what's unique is that sometimes those same proteins and pathways become dysregulated. And if they're not regulated properly, they can give rise to cancer. So the liver has sort of like two faces. One is good regeneration, which will make more liver when you need it. The other one is the bad regeneration, which is liver cancer. So it depends on if those pathways are regulated properly or not.
So there's one question in the chat, Andy.
Okay. What caused your transition in your postdoc? Do you mean when I left my postdoc or when I went to my postdoc? Or do you mean why did I switch things that I was working on? Any idea?
The things you were
Oh, the
working
things
on.
were, oh, great. Okay. Yeah. So when I was in graduate school, I worked on the blood and hematopoietic stem cells, how it is that they give rise to all the different lineages of the blood. So I actually, at the time, looked at 3 different postdoc opportunities. One was a lab that worked on hematopoietic stem cells. And then another lab was looking at embryonic stem cells. At the time, this was 2004 or 5, iPS cells had not even been invented yet, so that wasn't an option. And so one of the things I thought was that embryonic stem cells and hematopoietic stem cells, those fields were really crowded. There were a ton of people working on that. And the lab I ultimately went and worked on with the liver was doing some work with liver stem cells. And I thought, well, that sounds cool. And I don't know a lot of people working on that. Maybe that's an area that I could be successful in with a little bit of less competition. And ended up going there and I started working on liver stem cells. And you guys may have heard about this. A lot of times in a lab, you'll get 2 projects. One is maybe a very safe project, and another one is kind of a risky project. And hopefully one of those projects will work for you. So I worked on liver stem cells, and then I also worked on those very highly mature hepatocytes, the polyploid hepatocytes. And it just so happened that the work on the polyploid hepatocytes started working really well in my hands. So that's what I decided to pursue. So the question was, why did I change things I was working on? I thought it was interesting, but it was also a little bit of strategy. I thought that was an area that fewer people were working on, and maybe I could use that to some kind of advantage. A little bit of luck, a little bit of accident, I guess.
Any other questions, Dr. Duncan?
I just have a question about like deciding to switch labs. Like, how did you know switching like 2 years in was right for you, if that makes sense?
Yeah, so you know how we talk about, I show those pictures of life happens as you're doing your lab work. At the time when at the end of my second year of graduate school, when I knew that my first advisor was going to leave, we had a baby on the way. And the idea of, and our families are from North Carolina, where we lived at the time, the idea of moving across the country with a new baby on the way was absolutely terrifying. So we decided to stick around close to where our families were who could help us out a little bit. But at the same time, it was a big gamble on my part to completely switch labs at the end of my second year of graduate school. This happens to some people, and I've seen it now even as a faculty member. It happens sometimes to graduate students, and it's hard at first when you have to deal with it. But in almost every single case, people come out much stronger in the end. They don't usually waste time. They don't have a much longer graduate career, and they end up being very successful either way.
Great, thank you.
Yep.
Any other questions?
Well, if you guys end up having any other questions, you're certainly welcome to shoot me an email, and I'll be happy to talk to you personally.
Great. Thanks, Andy. All
Thank you.
right. That is it. I will talk to everyone tomorrow. If you need anything, please let us know.